Okay, I think we can get started now. Okay, so to recap, uh, yeah. So to recap, last class, yesterday, we talked about different types of sequencing technologies. Then we looked into different types of pre-processing techniques, and then finally different types of algorithms for sequence alignment. So today we'll take off from where we left off last class. So after sequence alignment, the part that comes is basically if you consider some sort of uh, 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 post alignment QC, and after that basically variant calling. So the post alignment QC is kind of optional depending upon the type of data you have. But yeah, we'll talk primarily about variant calling and different types of variant calling algorithms, including SNP calling and SV calling. Okay, so let's get started. And yeah, at the end of this session, towards the end, I'll show you a couple of examples and how we run the pipelines and basically do some hands-on stuff with the data, at least. Yeah. Okay. So let's get started. So firstly, why are genomic variants important? Why do we need to know about different types of variants inside the genome? The thing is, if you take two individuals randomly, if we are talking about humans here, if you take two individuals randomly, it's all about the genome is about 99.9% or 99.6 around that range, more than 99% similarity between two individuals of the same species. But there are these subtle differences, this 0.2, 0.3% differences inside the genome that make us who we are. So this is the reason the differences in the genome in terms of the variants are the reason because we have these distinct characteristics, distinct hair color, distinct eye color, distinct height, weight, everything. So these variants are actually pretty important to characterize the different kind of phenotypes that we generally exhibit for typically speaking about human beings. So we have you can measure any type of phenotype and kind of associate that particular phenotype with a particular genotype, as in this case will be some sort of SNP or SV. So this kind of analysis where you kind of associate a particular variant inside the genome with a particular phenotype exhibited by the individual or a group of individuals. By phenotype, I mean it can be anything. It can be height, weight. It can be lipid parameters like cholesterol, triglycerides, HDL, LDL, stuff like that. It can be other types of uh, blood biochemistry parameters as well, like red blood cell count, WBC count. So everything is kind of associated. There is always a genetic reason for a particular phenotype being high or low or even normal in some cases. So those kind of analysis associating a particular variant with a particular phenotype is done using GWAS studies. We will not go into details of GWAS, but I just wanted to mention GWAS and different types of association studies. So if you want to know further, there's a very good paper on GWAS in Nature. So it kind of just a second. Yeah. Uh, yeah. So kind, I'm not sure if this is the paper. There is another paper I'll find and I'll send you. Uh, mm,
Hmm.
okay, I'll send you at the end of the class. There's one nature paper actually where it very beautifully depicts what is GWAS, how our association studies done, what is the different type of data that we require for association studies, and so on. So it's pretty simple. You have a regression model. You have something of the order of y equal to mx plus c, where y will be your phenotype vector, x will be your vector containing all the variants of your interest, and somehow you find basically you run this model and find significant association of particular variants with particular phenotypes. But that is not the topic of discussion for today. We are delving a little bit deeper than that. We are trying to see how these variants are actually uh, uncovered from the genome. So, uh, what is this whole procedure of kind of uncovering different types of genomic variants from a sample or genomic data set is called variant calling. So as we discussed in the last class, so after the sequencing has happened, the reads will be generated and they'll be stored in a FASTQ file, and then we align those reads to a particular reference genome and create a BAM or a SAM or a BAM file, or in this case, GRAM files as well. And then we identify the different types of variants inside the aligned BAM files and write them onto a specific file called a VCF file, which is the variant calling format. We'll discuss about these things today a little bit in detail. Okay, so this is actually a good representation about how different types of reads are actually aligned to a particular reference genome. So this is your reference genome, and there are a lot of reads spanning I'm not sure if this is the entire genome or not. At least this part of the genome, there are a lot of reads spanning out. But if you see, say for example, in the first read on top, this A has been replaced by a C in the read, and this C has been replaced by A in the read. But if you look into the other reads that are there in that particular region, in the other reads are not supporting this particular change in in your sample. So the other reads are actually showing A and C, which is matching with your reference genome. So there also comes a point you have to consider there will be the different types of sequencing errors, base calling errors, everything can happen on that front. So that is the reason that you want to increase the uh, depth of the coverage of of your particular sample, because the if the number of reads supporting a particular base increases, if there is a variant, the number of reads supporting that particular variant will also increase. So you'll have more evidence to confidently say that there is a variant there, or if there is not a variant there. So, for example, in this case, out of one, two, three, four, five, six reads, out of six, only two are supporting that there is a variant, and all four are saying, and the rest of the four are saying that there is not a variant. So different types of variant calling softwares take these different types of evidences into account. How many reads are it is supporting? It is supported by, is there any other type of evidence present for that particular SNV to be called? And then if it gathers enough evidence, then it will call that particular position a variant, and it will give you the position and the reference. So for example, let's assume this is a variant. So in this case, the reference will be a T and the alternate allele will be a C in this case, right? So this is more or less how the variant calling softwares work. But yeah, there can be different types of variants. So the smallest genomic variants are the ones that I was talking about, the SNPs or the SNVs, the single nucleotide variants. It's basically a single nucleotide change. So it can be from a C to T, C to A, C to G, whatever what is a reference allele. The alternate will be something different to what it is actually there in the reference genome. So these are commonly known as SNPs or SNPs. Sometimes you'll hear people mention them as SNPs. So this is what SNPs are. So basically, these are single nucleotide differences inside the genome when you compare it to your reference genome. Right. So yeah. So these are three examples given here. So it's basically it's a C to T change here, and in this case there is no change in this case, and there is no change in this case as well. So in the first case that's an SNV, in the next two cases it's not. So basically there can be other types of variations in the genome which are like not single nucleotide changes. There can be massive, very big changes inside the genome. Anything about 50 base pairs change, it can be an insertion, it can be a deletion, it can be a duplication, or any other kind of genomic rearrangement that can happen. If the length of the base is number of bases for that particular rearrangement is greater than 50, we call that uh, structural variance. So we analyze structural variance separately, and we like kind of uncover structural variance separately, and we uncover SNPs and small indels. Like these are what you can call small indels, 3 base pairs, 4 base pairs, something like that. So these kind of small insertions and deletions, we club them together with SNPs, and the bigger ones, which are greater than 50 base pairs, we call them into a separate category called structural variants. And the algorithms used to uncover SNPs and structural variants are a little bit different, but again, the primary motive is same. All the variant calling softwares, be it for an SNP, be it for an SV, will always look for particular evidences that if there is at all a variation there, and according to the different evidences it collects, it will report it as a confident variant, or it will not report the variant. So the types of evidences that is used by this particular variant calling softwares is slightly different. So some software might use the depth of the region in which the variation is occurring. Some might use the paired end distance. So basically, if you remember when we were reading about Illumina, so it's a paired end sequencing. You are reading the same read. You are basically sequencing the same read from two ends. So one will be read one will be read two. Now, this read one and read two will have some distance between them when you are actually generating these reads. Now, if that distance is not the same when you are aligning the reads, then that means there is some sort of variation in the genome. So these kind of evidences, there are other evidences as well. We'll discuss them later in this class only. So these kind of evidences uh, are put forward by are looked for by the variant caller, and according to the evidences that it finds, it reports the variance. So yeah, now one interesting variation that occurs is basically tandem repeats. I hope many of you have heard this term. So it's basically short stretches of nucleotides,
हेलो
like yeah,
गुड इवनिंग सर हाँ जी
yeah.
नहीं नहीं मैं तो अपना बोरिया बिस्तरा बांध के रूम पर आ गई हाँ
Sorry,
जी
I'm muting you all. So if you want to say anything, please write in the chat. Yeah, I will discuss this once I finish this part. Okay. Yeah. Uh, Yeah, so these tandem repeats are short stretches of DNA that are repeated multiple times. So in this case, if you see, this is a tandem repeat, which are like short stretches of three nucleotides, CAG, and it is repeated multiple times. So different types of variations can occur. So here are four examples given. One is of an insertion, one is just a single nucleotide change, one is a deletion, and one is a tandem repeat. So these actually, if this happens for a long stretch of nucleotides, so this can happen, say for example, for 1,000 bases or 2,000 bases, then we call it a structural variation. Same with insertion and deletions, right? Okay. So the mutations that are occurring inside the genome, a person's genome, is can be of two types. It can be germline, it can be somatic. The somatic mutations are primarily those mutations that are acquired during one's lifetime. It is not actually inherited from the parents. It is acquired during one's lifetime, and that mutation is not passed on to the next generations. So diseases like cancer are very good examples of somatic mutations. In some cases, we have seen that some of the variants that are passed on from parents to the child are somehow linked with cancer and may increase the risk of cancer. But so the major kind of mutations that we see in genes like P53, genes like BRCA1, these are all acquired mutations during one's lifetime. These are somatic mutations. These do not occur in the gamete, so these will not be passed on to the next generation. On the other hand, you have germline mutations, which are basically inherited from the parents and passed on to the next generations. So these are uh, these are the most like the more kind of like rare disease kind of variants that are passed on in terms of germline mutation. So if you remember from your +2 levels, there's one example of a colorblind variant that is an X-linked recessive variant that is passed on from one generation to another. But it actually it's basically it's a rare disorder. So it's the most of the rare disorders that you see, most of rare neurodegenerative disorders, epileptic disorders, developmental disorders. These kind of disorders are mostly your Mendelian disorders, and these are passed on from one generation to another due to some mutations in particular genes or particular region of the genome. Right. So we will be focusing mostly on the germline mutation, not the somatic mutations, but obviously there are separate softwares for calling germline and somatic mutations. So if you are studying cancer and those type of diseases, you need to study somatic mutations more. If you are studying population genetics, how information is passed on from one generation to another, how these variants are passed from one generation to another, then germline mutations is the thing you should study about germline mutations, not the somatic ones. Right. Now, there are a few considerations to be dealt with while detecting variants. So, the variant detection step is actually a downstream step, considering that first you have to generate the sequence data, then you need to align it, then you need to do some QC steps, and before and after alignment, in some cases, then you need to do the variant calling. Now, since it is a downstream step, the upstream steps to this actually affect how well a variant calling algorithm will work, or how many or how well a variant caller can uncover variants from a particular genome. So a few important points to note, important factors that affect this variant calling is DNA isolation. Depending on how you isolate the DNA, how you uh, break down the DNA while you are uh, sequencing the DNA, it also matters when it comes to variant calling. We'll discuss it about these factors a little bit in the next slide. So bear with me. And PCR duplicates is one more reason is as well. We'll discuss why PCR duplicates is a reason. Coverage is a very important concept that we discussed yesterday. Is basically is all of your genome, every base in your genome covered at least once by a particular read. If, say, for example, your whole genome is not covered, it's 95% or 90% coverage you have, then the rest of the 5 or the 10% of the genome that you have will not have any supporting reads for that particular area of the genome. And if there are variants in the genome, obviously you cannot find those variants because these reads are what support the evidence for these particular variants to be found. And obviously read length and platform. So read length you mean either it is short read like Illumina is giving around, like, if you consider both the reads together, 150, 150, 300 base pairs around. And yeah, if you can compare it to other long read sequences like ONT, it will give you more than 1 KB. So if there is a structural variation, say for example, if it is 1,000 base pair long structural variation, it is very hard to confidently claim that structural variation is there in the genome if you are using short read data. If you are using Illumina sequencing, 300 base pairs of read length you have, it's very hard to justify if you are finding a 1,000 base pair long 1,000 base pair long structural variant. On the other hand, if your read length is say around 2,000 base pair long, and if you're finding a structural variant which is 1,000 base pair long, only one read is enough to support that there is a structural variant there, which is not the case for short read data as well. So it's always better to use to use long read sequencing data like ONT PacBio for structural variant detection. But yeah, it can be done using short read data as well. We'll do it later in the classes only. Okay, so uh, different considerations that we were talking about. So PCR duplicates. What are PCR duplicates? It's basically the same read. So when you are trying to amplify the read to get a better signal while sequencing, you're using PCR amplification, right? So if you are amplifying the read and by some chance or technical error, the same amplified read uh, is featured more than once inside in the reads of while aligning. What it happens is, say for example, this is on the right, this is a PCR duplicate. You are having the same read, but here it is depicted as two read, but it's actually the same read which has been duplicated. So there are actually two reads supporting that there is an A here, but there is one read supporting there is a G here. But technically it should be one versus one read supporting A, one read supporting G. But in this case, due to PCR duplications and contamination in your data, what is happening is there are two reads that are supporting uh, a particular variation. In this case, the two reads are not supporting that variation, and one read is supporting the variation. So it becomes difficult for the software caller, software for the variant caller to identify correctly a particular variation that is there in the genome. Another issue is basically non-coverage or less coverage across like non-uniform coverage, to be fair. So what happens is, say for example, there is a high coverage of reads here and there is a very low coverage of reads here. So what do you think is if there is let's not talk about coverage, let's talk about depth. If there is a high depth in a region inside, say for example, in a particular region in the genome, if there is the depth is quite high, it is say 5 times the non average depth of the whole genome, and there is another region in the genome where the depth is 5 times lower than the average depth of the genome. What does that actually mean? So if your depth is particularly very high in one part of the genome, say for example this part of the genome, the depth is very high, what it means is the reads that has been generated from these like region are are repeated in some way. So if you have repeats in the genome, so the reads that will be generated will be of similar sequence, right? So if you have extensive repeats in the genome, the reads you are generating from that portion of the genome will have extensive repeats. So when you're trying to align these particular reads to the reference genome, in the repeat region, your uh, depth will be very high. So that is an indicator for a variant calling software that there might be a variation here. On the other hand, when there is a lower depth, when there is lower depth or the lower depth compared to the average depth of the whole genome, then what it means is basically some part of the genome there is missing. So the depth is being compromised because the reads are not being able to align to this position, because that position was actually not there in the genome. So that means there has been some sort of a deletion or some sort of variation that has occurred there, right? So these are different types of these things that are playing a role in helping or kind of sometimes confusing a variant caller if there is a variant in that portion of the genome or not. But I just want to tell you that these variant callers are not very, like, these are actually sophisticated softwares. If you see softwares like Deep Variant by Google, and there are other variant calling softwares as well. So these the newer ones come with neural networks, machine learning stuff, and all those things. But the original idea behind the variant calling is to understand these different caveats inside the genome. If there is more depth, if there is less depth, how many reads are supporting a particular variation, or if at all there is if at all there are reads supporting a particular variation or not. The idea is similar. The way of doing things is different. We have evolved our way, computationally, we have come a lot of way forward. So we have newer techniques now. We have machine learning and deep learning algorithms now. But the major idea is to kind of identify this not so uniform regions in the genome, which can be depicted as some sort of variation compared to a reference genome. Now, one thing to remember, the reference genome that we are using, that we have been talking about from the last class for alignment, also the reference genome alignment is what we are looking at while we are calling variants. The reference genome is primarily a European genome. So the reference genomes that we have uh, available, say for example GRCS 38, or most recently T2T, before that GRCS 37, and so on. Even from the first Human Genome Project, the reference genomes that we have are mostly European genomes. So uh, systematically, biologically speaking, there will be like differences in in basically variations for compared if you compare a European genome with an Indian genome, for example, there will be inherent biological differences between these two different types of populations from different parts of the world because of different reasons. There can be like how migration patterns can affect these particular variations. There can be other factors, environmental factors also affecting variations. But there will be systematic biological difference between two genomes of two different regions of the world. So that doesn't mean that one genome is like has, say for example, if you keeping that in mind, if you are trying to align an Indian genome to a European genome, you'll find far more variance if you compared to if you align a European genome to a European genome. So that thing also you need to keep in mind. Where from is your sample coming? Are you supposed to see so many variations inside the genome, or is it actually the ancestry is very similar to the genome you are comparing it against, right? So these are considerations that you need to make while after actually doing the variant calling and doing the analysis further downstream analysis of the variants that you are finding. Okay, moving on. So this is kind of the uh, mm, uh, the pipeline that we'll be following for today's class. So we have FASTQ files. We'll kind of look into the different QC parameters using FastQC. We'll align it using BWMEM, which is a software that uses Burrows-Wheeler transform. So if you remember last class, we talked about suffix arrays, suffix trees, how indexing is done. So along with suffix arrays, uh, uh, BWA uses a concept called Burrows-Wheeler transform, which kind of is a compression technique that can index a particular genome. That if we have time in the end, I'll cover that part as well. So we'll align it using BWMEM, and GRC38 is the reference genome. So you can look it up in NCBI or uh, any you can just Google it, what is GRCS38 basically. And after this alignment part, so after aligning, the file that will be generated is going to be a SAM file, which is a slightly bigger and human readable file compared to a BAM file. It's BAM and SAM are actually the same file. BAM is the binary version of SAM. So we'll convert the SAM to a BAM because BAM is a smaller size of the same file and it's easier to handle. And yeah, and then we'll do variant calling. So we'll do SNP variant calling using deep variant. Deep variant is one of the latest softwares from Google. It's actually free. It uses this machine learning techniques to call variants, but the idea is similar as I was saying. And also we'll be calling structural variants using a separate software called Manta, right? So it's also a software that has been developed by Illumina themselves. So it works very good on Illumina data. So we have the data that we have currently is basically Illumina data. It's a human data and it's an exome sequencing data. It's not WGS, it's not whole genome. The introns and the intergenic regions are not there. It's just exome, the genic regions are present. And then we'll use the variants and we'll annotate these variants using different softwares, and we'll see actually what these variants are meaning. Right. So, yeah, we'll come to this later. Yeah, BQSR is something that I wanted to cover, but it's actually pretty interesting. So this step actually comes after alignment and before uh, variant calling. So what is BQSR? Or like, sorry, after variant calling and after alignment. So BQSR is basically you have when if you remember when you are calling the base there was a base calling software which basically converts the image of the sequencing to a particular base. So it is a software that does that. Now, if like while it is doing it, it also assigns a quality score to it, which is the Fred scale quality score that we saw yesterday as well. So it assigns a quality score to it. But what happens is after alignment, what can happen is you can understand that there are systematic errors, more errors inside the calling quality scores as well. So maybe something should have been more, it is less. Something should have been less, it is more. So what happens is the scores produced by the machines are subject to various sources of systematic technical error, leading to over or underestimated base quality scores in the data. Uh, some of these errors are due to the physics or the chemistry of how the sequencing reaction works, and and some are probably due to manufacturing defects. So this process, BQSR, base quality score recalibration, is a process that applies a machine learning model to model these errors empirically and adjust the scores accordingly. What does this mean? For example, say for example, for a given run, two A nucleotides were called in a row, and the next base had 1% higher rate of error. So what this software will do is basically if it will check in the whole genome where there are two A's together, and the nucleotide next to the two A's, the quality of that particular nucleotide will be reduced by 1%. So there can be other covariates, multiple covariates like this. Like one condition is like this: if there are two A's, the next base will have 1% lower quality. There can be other conditions like this. So uh, many of these covariates are additive in nature. So what will happen for one particular covariate, the base quality might go up. For the other particular covariate, it might go down. So it's an additive process. So after it is done, so it basically what it does is it adjusts the quality scores of the bases more appropriately so that it can help in further downstream analysis. Okay, let's come to structural variations. So after this, we'll look into the actual data. Sorry. Yeah, yeah, yeah. So structural variations, as I was saying, are variations inside the genome that are minimum 50 base pairs long. So that is how we define it. That is not actually what to say. That is not very biologically relevant, but due to our uh, uh, usage and our ease, we use this particular definition of minimum 50 base pairs, and we classify it into a separate category of structural variations. So this structural variation can be of two types. One is the copy number variant and another is the copy neutral variant. So the name suggests what it actually means. So when there is copy neutral variant, there is no increase or decrease of DNA in that particular region of the genome. So inversion is a very good example of a copy neutral variant. It's basically a part of this genome has been just inverted. If it was ACG, it's now uh, GCA. I'm talking about three bases, but this will be like more than 50 base pairs long, right? So this is one very good example. Another very good example is translocation. Translocation is basically cutting off the DNA from one part of the genome and kind of pasting it in another part. So the total amount of DNA does not change, but the region from which a particular DNA originates, that has changed. So this is also a structural variant, but it's a copy neutral variant because the amount of DNA has not changed. But in case of copy number variation, then the amount of DNA changes. Deletion, insertion, and duplication. These are the three major examples of copy number variants. So these are known as CNVs. On both are actually, if you use short form on both are CNVs. But by CNVs, we generally understand copy number variations, which are deletions, insertions, and duplications. So just one thing before I move on to the next topic. So we currently are we identify the CNVs or SVs, whatever you want to call them, using genome sequencing data, right? So we sequence the data and then we try to find evidences if there are variations greater than a certain number of base pairs inside the genome. But previously, this sequencing technique was not that was well established. So we had array designs. So remember microarray we talked about last class. So previously there were arrays which designed for a particular probe, and if that particular probe was higher in number, you could have assumed that there is a duplication. If that probe is lower in number from what you are expecting, you can assume there is a deletion, stuff like that, right? So you could actually identify copy number variations using array data. So previously, if you see some older papers for CNVs, you'll see they have done it using arrays. But right now, since the sequencing technology has been so streamlined and so easily available currently, so we generally use sequence data only to call structure variations, even CNVs. Okay. Now, as I was saying, there are different types of evidences present to kind of identify this particular structural variations inside the genome. So as I was saying, there are if you are using Illumina, it's paired end data. So that actually comes in very handy while detecting structural variations. Say, for example, you have an insertion in place of the genome. So what you'll have is two paired end like this, paired end reads like this when you have generated the data. So you know that these two reads have a minimum distance of something between them, right? It's two paired reads, and you already know the distance between the reads in your original genome. Now when you're trying to align the genome, what you see that these two reads, after aligning, the distance has increased between these two reads, right? So what does that mean is, inside between these two reads something has been inserted. That's why the distance between these two reads has increased. So this can act as an evidence. This is a paired read evidence or a paired end evidence for detecting an insertion. So for an insertion, if you have paired end data or paired end reads, if the distance between these two reads increase after you have aligned your genome to the reference, then it is indicative of an insertion. For deletion, it's actually opposite, and it's intuitive. If there were two reads which had previously 50 base pair distance between them, and after aligning there is two base pair distance between them, that means there is a 48 base pair deletion inside the genome. So these two reads will come more closer to each other. Uh, closer to each other when there is a deletion after sequence alignment. So these are known as discordant distance between paired reads. This method of identifying structural variations using paired reads and discordance between them is called discordant distance between paired reads. So that's the parameter that is used to quantify this. Now, next we come to duplication. As I was saying, for duplication, that if your sequencing depth across the genome is not same, if there are places where the sequencing depth is way too high, if like normally if you have 30x 40x in one place, if it is 80x, then that's not normal, right? What that is indicative of is basically the same read has the same region of the genome has produced sorry, different regions of the genome has produced the same read. What it means is actually there is duplication inside the genome. It's the same read that has been produced by different areas of the genome. That means that whole area has been duplicated. So the depth will dramatically increase in case of sorry, in case of duplications. And uh, in case of deletions, again, if you want to see deletions, the depth will dramatically decrease at that particular area of the genome. Now, translocation is a very interesting SV that is detected using split reads. So, what are split reads? Is basically, say for example, you have two reads, or yeah, let's assume that you have only one read that is that has been generated from one part of the genome. Now, what is happening is when you are doing sequence alignment, what you are seeing is the whole read is not aligning to one particular area of the genome. One part of the read is aligning to somewhere else. The other part of the same read is aligning to somewhere else. So basically, your same read has been split, and half of it is or half or less than half of it is aligning to some place else, and the rest half has is aligning to some place else. So what it means is a particular area of the genome has been rearranged. That particular area of the genome has been rearranged. Maybe some part of the genome has been cut and pasted in that area where the rest of the read is getting aligned to. Right? It's intuitive. If you think it like that, you'll it's easier to understand. So you have a whole read, but the whole read is not aligning to your reference genome at one particular region. Half of it is aligning someplace, half of it is aligning someplace else. So what it means is, from where the read had been generated, that region, half of it, or half of the region has moved to someplace else. That is why half of the read is aligning to someplace else. So this is also very interesting, and this is how different types of uh, software called variant calling algorithm softwares use these particular evidences to claim that there has been a particular uh, translocation or an insertion or a duplication or a deletion, right? And inversion, I missed out on inversion. Inversion is also pretty interesting to notice, is because if there is an inversion, if you had opposing reads, if you had paired reads which are opposing, what will happen is you won't have these opposing reads. You'll have two reads, but both will be facing towards the same place, towards the same area. So basically, if it is one read, you had generated 5 prime to 3 prime, and another you had generated 3 prime to 5 prime, but after alignment, you see both of them at 5 prime to 3 prime. That means that area of the genome in which the other read has been inverted, there has been an inversion in that particular read of area of the genome. So all of these evidences that we use to detect SVs are based on sequence alignment. How well your data is aligned to how well your sample genome is aligned to a reference genome, and what are the different caveats of this alignment. Is it aligned too much? Are there too many reads there? Is there less number of reads there than expected, or something else? So different caveats and different these minute changes in alignment is the evidence for variation inside the genome. Right. Okay. So before we talk about VCA format, I want to talk about a little bit more about these SVs. So uh, there are certain terms that are used for like for these particular evidences. Okay, yeah. So you can have simple structural variants and you can have complex structural variants. What is the difference between these? So simple structural variants is basically one variant, one type of variation in that particular area of the genome. So it can be just an insertion. So for example, this is a reference genome. In this case, this is an area A and B. So in this case, A has been duplicated, or B has been inverted, or say for example A has been deleted, only B is there. So these are like single type of variations in that particular area of the genome. But it can also happen that there might be multiple things happening together. Say for example, in this case, mm, excuse me, in this case, deletion and duplication has occurred together. So how this A region has been deleted and the B region has been duplicated. So these are what are known as complex structural variations, where more than one type of variation is occurring inside the genome, right? So yeah, these are the different kind of methods or evidences that are used by particular variant callers. So on the right, you see this table. These are your different algorithms or tools that are used. Like many have been developed in the recent years. So Delhi and Lumpy are quite widely used on WGS data, and breakdancer and CNV-nator. CNV-nator, you see, CNV-nator can work on array data as well. So if you have array detecting CNVs from arrays, CNV-nator it can work on that as well. So there are different types of these variant calling softwares, and they use different types of evidences to call variants. So these type of the evidence type can be segregated into four major evidence types, right? So the first is the read pair methods. So as I was saying, in Illumina, you have these paired reads. The distance and the orientation relative to these particular reads can be used as an evidence to claim a structural variant there. As I was saying, for example, for inversion, if you had two reads facing each other, after alignment, you'll have both the reads facing towards the same side. Right. So read pair methods actually look into these paired reads and their orientation and the distance between them prior prior to sequence alignment and after sequence alignment. If there are changes between them, then there is a high possibility that there is a particular variation inside in that particular area of the genome. The next method that is used is read depth or read count. As I was saying, how many reads are going and aligning to that particular area of the genome. In general, we assume that it's going to be a Poisson or a modified Poisson distribution in mapping depth. And if there is some divergence from that particular distribution, then there is high possibility that there are. So for using read depth, we generally only identify duplications and deletions. So if there is a shift from a Poisson distribution or a modified Poisson distribution to a to something else, or there's you can see there's a clear shift in the distribution, then there is a major possibility that that particular uh, region of the genome has a deletion or a duplication, right? And the third method is split read methods. As I was saying, the same read that has been aligned to two different places of the genome. It's the same read that has been split and aligned to two different parts of the genome. Mostly we identify translocations using split reads, but deletions can also be identified, or insertions can also be identified using split reads. For example, in insertions, if a part of the genome has been inserted, the left of region of the genome of left region of the insertion and right region of the insertion, they were actually together. They are supposed to be together, but instead of that they has been split, and some sequence of nucleotides has been added in the middle. So what happens is the original read that had been generated from the sorry yeah, the original read that should have been a single read, the single read is been split into two different reads. Like this, you can identify these deletions or insertions using split reads as well. So these are the major three methods to be major three evidences, as I may say, used by these different types of structural variant callers. But there is also a fourth method, which is the de novo assembly. It's basically I did not talk much about the genome assembly techniques. So say for example, if you are I've been mostly talking about human data and like known species data. But say, for example, you are sequencing a species that does not have a reference genome. What are you going to do? So you need to assemble the genome from scratch, right? From these particular leads, you need to assemble the whole genome. So the generally this genome assembly technique in this particular type of assembly, what they do is they you have these particular leads, you find overlapping sequences between these particular leads, and you create what is known as a De Bruijn graph. I'll not go into the details. So it's basically a graph where you have these nodes and edges. Each of the node represent these k-mers. This k is basically the length of the sequence of the leads. So if it is a like a 10 base pair read, it's a 10 mer. If it is a 2 base pair read, it's a 2 mer, like that. So these individual nodes of the De Bruijn graphs represent these k-mers, and the link between two particular nodes, the edge between two particular nodes, represent the overlap between the two reads. So like this, you can create the whole graph using these nodes and edges. And after that, how will you find the optimal genome assembly is something called Eulerian walk on the graph. So basically, I'll not go into the details of Eulerian walk. It's basically you start from a node and you kind of traverse the whole graph depending on node to node connectivity. So if this node is there's a node, there's another node left to it, and there's only one connection between them. So you move from this node to the next node. If from the next node there are two connections going on to two different places, so you then use probability measures. So 50% of the time you go left, 50% of the time you go right. So I'll not go into the details of this techniques, but the basic idea is you create a graph, and basically you traverse the whole graph to get the original genome, or original string or genome, whatever you want to say. Now, in case of structural variant calling, this kind of techniques are also used. These are known as de novo assembly. So what they want try to do is basically assemble assemble the original sequence from the short fragment of reads, and basically then try to see where there are where in the genome, the particular region is not matching with the reference. So primarily these four techniques are used, mostly the first three. The fourth one is a little bit more sophisticated. So Manta, the software that I was talking about, uses all four. So it's actually pretty good, and it uses all the four of the techniques, and it gives you a consensus of all the four of the techniques, and gives you a confident uh, uh, variant call. Now again, like when there was a base caller, and the base caller was identifying different nucleotides from the sequence uh, during sequencing, it had a probability of error associated with it. It had a quality score associated with. Same with these types of variant callers, it will give you a probability of error or a quality score associated with a particular variant to tell you how confident they are that that particular variant is present in that region or not. Right? Okay. Yeah. So yeah, let's talk about the VCA format now. So this, I am not going into details of this, but this slide is also you can see intuitively what these different reads are actually telling you about these variations. So for example, the major difference between short read and long read, as I was saying, while detecting structural variant, is basically only one read is enough to kind of tell you that there has been a variation inside that region of the genome if the variation is like smaller than the read length itself. So in case of long read, you'll have 2000, 3000 base pairs, even more. In some cases, you can have 10 KB base pairs long reads as well. So it can easily identify these structural variations, which are mostly around, if you sample all the structural variations in the human genome, the average size you'll find is around the 1000 mark. 1000 1200 mark, right? You can have extremely large, you can have like thousands of bases that have been duplicated, and you can have smaller number of bases that have been uh, mutated as well. But on an average, it's like around the 1200 1300 1400, so that range, right? So long read data provides much better confidence in calling these particular variants because one read is necessary to tell you the whole story, right? You can have multiple reads aligning to that, it will give you more evidence. But if one read is telling you all you need to know, that's actually pretty good, right? So that is why we actually prefer long read sequencing when we are trying to call structural variants, right? Because the read length is more, the information that it can encode is much more. Okay. Okay, so let's now talk about the VCF format. Like yeah, after you have called the the variants, how are the variants stored in the file, right? So this file format is known as VCF format. It stands for variant calling format, and it's a very commonly used data format for variation variation representation, right? So these are basically ultimately at the end of the day, these are tab delimited text files, but there is a catch to it. On the top, you have a lot of headers which actually tell you about what different terms in the file mean. There will be short forms. Say, for example, AA. What does AA mean? What does AC mean? AC actually means is allele count. Allele count. So how many times this particular allele is there for that particular variant, right? So these full forms and this information about what these particular terms mean and what are the different types of commands that has been undergone for that particular VCF file to be generated, those information are there all in the header, and it also tells you the VCF format version. So currently it's around 4.1, 4.2. So they keep on changing a little bit, and they release another version like that. But yeah, it's essentially in the important part in the variation, seeing the variations and like listing the variations, that part is pretty straightforward. It's tab delimited file, right? So on the first column you have the chromosome. Next you have the position of base pair position. This ID is basically if you have if this particular variation is priorly known, if it is already been reported. So this RS334, you see this RS ID is corresponding to a particular SNP, and the ID is corresponding to a DB-SNP ID. So the DB-SNP is a database of single nucleotide polymorphisms. It's a database hosted by NCBI. You can just go and take a look. And in that database, so every variation uh, is given a particular ID, and that starts with RS, and then followed by a number. So this is what the ID means. And ref and alt, these are clear. So ref is basically your reference allele, what nucleotide is there in that particular position in the reference, and what nucleotide is there in that particular position in your sample is given by the alternate allele. So in your case, it's A. So there has been a change from T to A in chromosome 11 in this particular position. So this is what it means, right? And then there's the quality parameter, as I was saying. It's a Fred scaled parameter. It's a logarithmic, negative logarithmic of the error percentage of of, in this case in particular, is calling this particular variant that there has been a change from T to A. So in this case, call is 100. 100 is a very high number, so you have like very low percentage of error. So it's actually a pretty good variant. And filter is something that a particular software assigns. So Manta will assign if it is passing, or so it will have some of its QC parameters. Say for example, Manta, as I said, is a software that uses all four evidences to claim that a particular variant is there. So what it will do is it will check all the four evidences and it will come up with a cumulative score, and it will tell you if all the evidences are supporting it, or if it is only one evidence supporting it, or two evidence supporting it. According to that, it will give you certain categorization. So in this case, it's pass. In some case, it will be low depth. That means there was not enough depth to identify that, confidently identify that particular variant, but it has been identified, but it's not confident enough. It can be low depth, low call, anything can be there. The pass is basically it passes all those threshold parameters to claim that this is a confident variant that has been identified. And after that, in the info column, you get all the supplementary information related to that particular variant. So AC, as I said, is allele count. AF is basically allele frequency. How many times has this particular position been mutated across all the samples you are checking, right? So you'll generally not be checking one single sample. You'll be checking basically a list of samples that may be like 50, 100, whatever, 10,000 as well. So out of those hundred samples, say for example, in two samples you find this position that has been mutated. So 2 out of 100, it's basically 0.002. It's that is the allele frequency of this particular position and this particular allele. So that is what allele frequency means. Uh, mm, uh, out of the whole samples, all the samples that you used, how many of the samples have this particular variant? That is depicted by the allele frequency. Now, if you see after that, AFR, AF, AMR, AF, then other things. So what these means are, so there are cohort studies that has been performed. So let me actually, uh, like this. Okay, so one of the cohort studies that has been performed is Nomad. Right, this has kind of done, I think, 500,000 genomes. No, I think UKB is 500. Nomad is also a very big number. I don't remember the exact number. Here you can, this is the database for Nomad. So they have all the information. So they have collected samples from all, like not all ethnicities, but they have tried to collect from all parts of the world and checked for certain frequencies of these allele differences between different ethnicities. Say, for example, in chromosome, let's check for that position only. So this is actually chromosome 11, 5248232. Right. Chromosome 11, 5248232. So this is the position. No, I don't think I put 5248232. Yeah, there is no. So this is just a dummy example. Let me. Uh, Yeah. So in this region, this 5248 822, 5248 and 5248262, meaning like 40 base pair changes. So there are these uh, documented variations that can happen. So in this position, there has been a change from A to T. And if you click on this, it will take you to this page and it will tell you which ancestry group has this particular variation. So in this case, only African American ancestry has this particular allele count. So there are two allele counts, two samples have this particular variation, and what is the frequency of this variation, right? So this is actually like pretty well documented. So you can see, say for example, another one, this one. So this is actually present across all types of genetic ethnic groups in nomad. So other types of these, like these are actually cohort studies which collect samples from across the world, in some cases across the country. So in our case in India, there has been a there's the Genome India project that I am also currently working on. So it actually collects samples from all over the country. We've collected 10,000 samples from all over the country, and we are also trying to make these kind of estimations to what is the different allele counts, allele frequencies, allele variations inside the genome, and if they are specific to particular population groups, right? So these type of cohort studies, nomad, 1 kgp is another. It's the 1,000 genome project, if you have heard. uh, Actually, in 1 kgp, if you want, they have the FASTQ files and everything you can access. So this is the website there. I hope you can see my screen, and yeah, I'll send you these links in the chat. So these are cohort studies that have occurred, that have documented this type of variations. Okay, sorry, I did not see the chat. Oh, yeah, it's already been answered. So, okay. Yeah. So these type of cohort studies have actually studied these particular variations and kind of documented them and how much of these variations are present in particular population groups. So we fall under the South Asian population group. So uh, this population group is our, right. So the Indians and other South Asian countries. So due to genetic ancestry, due to the origin of your ancestry, there can be these variations that can be different from a person with having a different type of ancestry. So billions of years of this admixture, different types of population groups kind of marrying and then having children, kind of passing these individual variations to their children, this has like brought about a lot of allele frequency changes in across all the humans in the population. So if a certain particular group practices endogamy, it's just like marriage within the same community, what happens is basically the same type of variants are passed on from the parents to the children across generations. So that is actually typically not good to do, because if there are disease alleles or if there are risk alleles, if there are variations that actually affect your phenotype or your individual disease or anything else, if that is passed on from generation to generation to generation, that's actually not good for the community itself. So yeah, I'm digressing, but yeah, you can look these up, and these are actually pretty interesting population genetic stuff, right? Okay. uh, yeah, so I'll not go over these because I'll show you the VCF file itself. You can have a look, take a look and understand. Yeah, so these are your basically all the parameters that has been listed in these particular headers. So you have chromosome position, ID, reference alternate, quality, and then you have filter and info, which gives you the extra allele frequencies information and everything. The format, oh, I didn't cover the format. So basically the format tells you, in this case, the genotype. So in this case, it's a 0, 1. It's a heterozygous genotype. That means one strand has the variation, the other strand does not. So this is a heterozygous genotype 0, 1. You see the representation here, 0 straight slash 1. So in some cases, you'll see 0 and a slanting slash then 1. So the major difference between a slanting and a straight slash is basically, if there is a straight slash, it means the data has been phased. What does phasing means? Phasing is actually determining if the genetic variant came from your father's strand or your mother's strand. So in your DNA, you'll have two strands. So one you have inherited from your father, another from your mother. If you can phase the data, having information from your parents as well, genetic data as well. So you can understand where from that particular variant has originated. Is it coming from the father's strand, or is it coming from the mother's strand? So if you have identified that information, it is given by a straight slash or a pipe, if you say. So in this case, it's a phased genotype. In some cases, you'll see a slash, meaning you know that it is heterozygous, that one strand has, the other does not have, but you don't know if it is coming from which of the strand. that information you don't have. So yeah. So this is how the VCF format looks like. And in the final step, after you have gotten the VCF, you have gotten the variations, now you would want to look into the variations. You would want to further analyze the variations. What are these actual variations? Are they affecting protein coding regions? Are they affecting splice regions of genes? Are they affecting introns? Or are they actually uh, 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 causing any particular risk to a particular disease? So these kind of information you would want. So there are databases that actually, first of all, there are softwares like this software is called ANOVAR. So what it does is it incorporates information from a lot of databases, and if that particular variant has been prior associated with a particular disease, it will report that that particular variant is a disease risk variant. So these kind of information, this further downstream information of these particular variants that you have uncovered, you can uncover it using variant annotation softwares. ANOVAR is one of them. There are more conventional ones called SNPF and VEP. So there are other softwares there as well. But yeah, this is the idea. Okay, so this, if you recall, we started from the sequencing data generation. Then we studied a little bit about pre-alignment QC, quality control, data trimming and those stuff, adapter trimming. And after that, we studied sequence alignment. After that, variant calling. What is variant calling and how do you call these variants? And once you have got these variants, how do you further understand these variants and using some medical information? Okay, if it is actually a protein coding region variant, is it actually affecting some sort of protein formation, or is it like actually not harmful at all? So in one thing to keep in mind uh, while looking at these variants is basically you are not supposed to find multiple disease-causing variants in across different samples. If you are taking 100 samples, it is highly improbable that out of these 100 samples, you'll find 90 samples which have particular disease-causing variants. Because the answer is pretty simple. It's because of evolution. Because if these variants are actually disease-causing, evolution from an evolutionary perspective, you wouldn't want these variations to be further passed on and these variations to be incubated in the population so that actually the quality of the population goes down and there is a lot more diseases. So evolution from an evolutionary perspective, this is actually counterintuitive. So actually what you'll see is very few individuals will have this particular disease causing variants, but the total number of variants that you'll see is very high. You'll see like billions of variants. It can happen like you are seeing like 250 million variants in a particular genome out of 3 billion base pairs. Now what it means is most of the variations that are occurring are low impact variations. Basically, they do not impact your protein coding or your any essential mechanism of the cell that much. So that's why those variations are surviving, and those variations are giving you distinct phenotypic traits, but they're not kind of like bad, say, like those are not disease causing, or those are not kind of yeah, those are not primarily disease causing. Those are giving you specific traits that is distinguishing you from another individual, but those are not always disease causing. So disease causing SNPs or SNVs or SVs you'll find are very rare in the population. So the allele frequencies that we were talking about here, so this allele frequencies, these AFs. So if we classify these allele frequencies into three regions. So one is greater than 1% of allele frequencies or greater than 5% of allele frequencies. Those are common. 1 to 5% of allele frequencies variations are uh, rare, and less than 1% are ultra rare. So like that, we quantify these alleles and check how are these allele frequencies distributed in different population groups. Right. Okay. I think yeah, I have covered in this part. I think the lecture part is covered. Now let's look at some data actually. Okay. Okay, I hope you can see my screen. So, I had written in my poster that I'll cover some Linux commands, but that was primarily for the fact that we'll use this genomic data analysis part, like we conduct this analysis part in Linux platforms only. But of course, it's not possible for me in a 2 hour class to give you a whole overview of Linux and stuff. But a few commands I'll say. So now one very easy command is cd, which is change directory. So cd, and then you put the name of the name of the directory like this. It changes the directory. ls is the list command, which kind of lists all the files in the directory. Now you can play around with it. You can list it and kind of sort it by time and stuff like that. So it tells you this is sorted by time, gives you all the kind of the size of the files that are present there. Now, say for example, you want to view a file. Say for example, I want to view. Uh, Say for example, I want to view this particular file. So this is a CSV file. The command for viewing that file is vi, or vi is an editor. So vim is the editor. It's a file editor in Linux. So you can use vi or vim to edit particular files, text files, CSV files, whatever files there is. Vi is the editor. If you just want to see the file, you can use a command called less. So if you type less and the name of the file, it gives you the file like this. And if you do vi, it will open like this, but here you can edit. So after you have gotten into vi, you can click on I. So if you see on the below, there's this insert thing that has come on. So now you can change annotation, like whatever you want to edit, you can edit. And after editing, pressing escape and colon, you see the colon has come here. The command for saving the file is wq. So if you do WQ, it will save the file. If you say, for example, do just colon Q, it will quit without saving, right? So these are like very basic commands that we use in Linux. And one important thing that we do is basically, since our systems are not well suited, actually, the laptops that we generally use in our daily life, they are not very well suited for handling such big data sets like genomic data sets like these big. So we often use some sort of high performance computers. So in our case, in my case, I use an HPC that is there in my department because like my laptop cannot handle this like heavy, this big files of data and work computations on those. So what we do is we connect to a remote Linux server, and which is there inside. In case, in my case, in my department as well. So there are other yeah, you can connect to any other Linux computer, not even an HPC. You can connect to any other Linux based computing system using a command called SSH. Then you need to provide your username. This is my username and the IP address. So in my case, this is the IP address right here. It asks for my password. I give my password and it does the login. So I have logged into a remote server, and I can access all the files. I can run commands on the remote server from this particular terminal like this. Okay, so I actually have already made all the files that is required, because the file that I have is actually a pretty big file, so I couldn't find a smaller file here. So the FASTQ files I have are more than 3 GB in size for individual FASTQ files. So basically, I cannot like if I run this in real time inside the class, this will take like just the alignment part will take like 18-20 hours. So it's not feasible for me to show you live running it, but I'll show you the commands. I'll show you the input, what the input looks like, what the output looks like, and I'll show you what are the commands and how to use them. So first, let's get started. This is the sample. These are the two samples that are there. So this is one FASTQ file. This is the other FASTQ file. So since these are Illumina generated files, first of all, these are exome sequencing files, as I was saying in the early part of the lecture. Okay, these are uh, Illumina generated files. So it will have two FASTQ files, one for read one another for read to, right? So let's just have a quick look about how the first few files look, right? So I'll use less because VI is an editing command. So if you use VI on very big files, it'll take a lot of time to load. So since we are not editing these files here, I'll use less. And so these are already, you see, these are, yeah, no, so uh, I think font size, yeah, I can increase that. But yeah, no, RNA sequencing is a slightly bit different because if you think about it, if I'm talking from a human perspective, the RNA, the mRNA that you are trying to sequence, that's actually not fully like present in one place in the genome, right? So what happens is from the genome, uh, RNA polymerase comes and makes the mRNA. But after the mRNA is formed, you do some splicing mechanisms on it. So the introns are cut off and then the exons are joined. So the information, if you sequence mRNAs, what the information that you'll have is only of the exons. You do not have the introns in between. So the alignment algorithm there works a little bit different. So there, what happens is the alignment needs to be splice aware. So in that case, a read in most cases will align to different parts of the genome because uh, those different parts are actually the exons in the middle part where there is no read. That's actually the introns. So those type of carriers you need to keep in mind. So I think I have one slide that I can show you for RNA sequencing. Uh, Yeah, so this is called star alignment. The star is a software. So basically, you have this read. This read can map to multiple regions. So there will be one. So these are your exons. The module ones are your exons, and the line connecting these exons are your introns. So a part of the read will align to one side of the exon, and the another part of the read will align to one side. So this is your donor side, so read one will align here, and this is your acceptor side when your read two will align here. A part of the read one will align there. So these type of things you need to keep in mind when you are doing RNA sequencing, because RNA sequencing is slightly more like what to say, it's not, I'll not say it's more difficult, but it's a little bit more uh, no, I'll not like I do not want to say complicated, but the thing is there are certain things that you need to keep in mind. Say, for example, this, and there might be like so RNA you cannot sequence directly, right? You cannot use RNA polymerase to sequence the RNA. So what you need to do is convert the RNA back to a DNA using a reverse transcriptase, and then sequence that complementary DNA, the cDNA you need to sequence. So there are some things that are different, but the most of the things are mostly similar, but there are some of these small things you need to keep in mind when you are doing RNA sequencing, and it's not the same software. Uh, Okay, so you're asking how do you integrate multi-omics data to enhance accuracy of GWAS? So I have not discussed GWAS much in this class, but okay, I'll try to answer this question. So in GWAS, what you do is you kind of associate a particular variant with a particular phenotype. Now, this particular association, you can have multiple types of data in between this particular association. So this on a primary basis, on if you do just GWAS, it's basically you have a SNP data, what are the SNPs in the sample, and what are the phenotypes of the particular samples. So you kind of try to use a regression model to, using a linear mixed model, you try to associate these particular two things. But there can be other evidences of this association that are not just genotypes. There can be gene expression data. There can be other types of data. Gene expression is one of them. There can be proteomics data as well. You can integrate those data inside this model to account for this variation in a better way. So integrating multi-omics data is actually pretty pretty state of the art, if you want to say like that, uh, to kind of associate these particular genotypes with phenotypes. But yeah, I have not discussed yours, so I'll not go into much details of it. So yeah, if you want to know more or if you want to discuss more, you might email me and we can discuss it later after this thing gets over. Okay. Yeah, I'll try to increase the font size here. Let me see. I hope now it's okay. Yeah. So this is how the FASTQ file looks like, as we had discussed. So this one is telling you it's coming from read one, and this is your actually your sequence that has been found. There's the addition sign saying meaning like the sequence is connected to the next sequence, and this is your ASCII coded this F string of Fs and these fives and dashes. These are your ASCII coded quality values for each of these bases called. So for C, the ASCII coded quality value is F. For N, in this case, there is no base there. There's an N here. So in case of N, there's a hash sign, and therefore other cases that you can find the like other types of quality scores as well. So this is how your FASTQ file looks like. So it's a very big file. I'll not go in to the full file, but this is how the heading of the FASTQ file looks like. Now the same thing is this I showed you for the read one file. You can see the same thing for the read two file as well. Uh, just the only difference there will be it's from the read to, it's telling you it's coming from read to, but the rest of the stuff is same, and it'll tell you the sequence, it'll tell you the quality scores associated with it and everything like that, right? So these are the two files that we have. So these are your raw data, right? Now what you want to do is basically check for fast QC. You would want to run fast QC on this. So what does fast QC do? If you remember from last class, so let me go back here. Yeah, so it assesses your different quality measures, and it gives you plots and different types of measurements. Okay, how much is there a deviation from your GC content, if there are duplications in your genome, or stuff like that, right? So let's see an example here. So yeah, so this, not this one. Yeah, this is actually the website from which you can download FastQC. FastQC is pretty easy to use. You can download it. It'll come in a zip file, and in the zip file, you'll find the executable file which you can run along with the FastQ file there. So I'll show you the command. So in since I'm using this in an HPC, I cannot use the command line directly. I have to form bash scripts like this .sh scripts, which will execute the thing in a queue. So I have to submit it in a node, and that command will be executed in a queue. So if I show you the command, so the command is pretty simple. So just like do not just ignore the top heading line. So this is just information about the command I'm trying to run. Okay, like how much time I want, what is the error file I want, output file, those stuff. So this is actually the command. So in this part, so fastqc/fastqc, this is I'm pointing towards the executable application for fastqc, and this is my output directory. I have specified using -o, and -t is kind of telling you, basically telling the function software how many threads to use in that particular node. So I'm using 20 threads, and these are my fastq files. So this is my first fastq R1, and this is my next fastq, which is R2. So I I actually do not have this particular FastQ result here, but I have some other FastQ results. I'll just quickly show you. Mm, not this one. Uh, just give me a second. I think, yeah, it's in this PPT actually. Mm, yeah, yeah. So this is how the FastQC report will come. It will come as a .html file like this. You can see the sample name has been given. This is for a different sample, but just I wanted to show you. So yeah, these are the different characteristic plots that will give. So this is the basic statistic: total number of sequences, total number of bases, and what is the sequence length. It ranges from 35 base pair to 151 base pairs, and your percent GC content is 51. Then you have different plots. So this plot is showing the quality scores of all your sequences. Everything is in the green zone, which means on the higher zone. So it starts from 0 and goes up to 40. So if it is between 25 to 40, it's actually pretty good. So your quality scores are actually pretty good. No issue with that. Per style sequence quality, what it actually means is, I've written it here. So it basically means that every flow cell tile, the reads generating from every flow cell tile, is it good quality or not. If it is actually full blue, it means it's actually good quality. If there are reds somewhere, it means that portion of that particular tile, the data that has been generated from that particular tile, is actually not good enough, right? So yeah, so you can see slight dots of light blue and red, which means like these tiles were some iffy, but the rest of the tiles, it's okay, right? Then you come to per sequence quality scores. So these are your Fred quality scores, as I was saying, on the right, and these are a number of sequences. So for maximum sequences, you have quality scores on the higher end, not on the lower end. Per base sequence content here is some like it's not exactly what you would expect to be, but you cannot term this as a sequencing error without further evidence. So we might not be able to do anything with this. Per sequence GC content, see, the theoretical distribution is a Poisson distribution, and the GC count per read is actually slightly deviated from your Poisson distribution. This is indicative that there might be some sort of contamination, but then again, this is no proof. So, the species might be like that only. Like your genetic makeup might be like that only. So it's no proof that your data is of low quality. So here you do not have any n per base in content. N is basically if there are any missing bases or not. So there are no missing bases in this case. And for sequence length, you have maximum of the reads, which is like 10 to the power 7 reads of like 150, around 150 base pairs. You have very low number of reads, which are actually smaller in size. So 80 to 90, 100, around 100. These are actually pretty low in number. And the maximum you have is around 150. So yeah, sequence duplication level also, you have like most of the sequences are not duplicated here. There is no overrepresented sequence, as for the example that I was showing in the PPT, there were duplications. There were duplications and overrepresented sequences here, but in this case, in our file, there isn't. So, sorry. So yeah, and this tells you the adapter content of the reads. So it has the adapter sequences for Illumina adapters, 3 prime, 5 prime, and other types of poly A, poly G adapters, and it did not find any kind of adapter in the data, right? So it pretty much means that your data is actually pretty good to go. Your read quality is good, your sequence length is your read length is good, your data does not have much of contamination and stuff, so your data is good to go for your further steps of analysis, right? So first step is doing QC, checking for QC, and the second step is like if our data had issues, if our data had adapter content or some overrepresented sequence, we might need it to trim those down using softwares like Trimomatic. Like, okay, I think I discussed mentioned Trimomatic a little bit yesterday, but again, not opening for some reason. Yeah, so this is the Trimomatic software. You can take a look. It is used to trim data. Okay, guys, I'm not following the chat currently. I'll look into the messages after I'm completed, right? Okay. Yeah. After you have done fast QC, now you would want to align the data to a particular reference file. So my reference file is is present in my home area, and this is the command that we use to run BWMEM, right? Sure. So again, so this is where my original BWMEM folder is there. I call that folder and call the executable there. And these, in the first inverted comma, you see these are your basically read group information, sample information. So the TSM is your sample in ID, and this is your, so basically this is your ID and this is your sample name and this is your platform, Illumina. So this is what it goes into. Hyphen T is 48 is basically telling you the number of threads to use. K equal to 20 is, as I was saying, the K merge, the read length. So it will only try to align 20 base pairs at one time. It will not try to do more than that. So this K value is actually it actually fluctuates if when you use WGS compared to when you use exome sequencing. So for WGS, this K value is higher. It's on 200, 500 range, even 1000 range. But in case of exome, you need to have smaller alignment regions because like your exome data itself is not that long, right? And after that, I give it the FASTA file. So this is my reference file. Hey, this is .fna.gz is basically .fna is the FASTA file of the nuclear, sorry, not nuclear, nucleotide format. FNA represents the FASTA file of a nucleotide, right? So this is your, this is my FASTA file, VRCS30. You can download this from the NCBI website. This is the latest version for VRCS30. Then I give the two FASTQ files, the full paths of the two FASTQ files. This is read one here, this is read two here, and then I pipe it to the output, and the output is a .sam file, right? So align.sam will be your output, and your input will be FASTQ. So this actually is the BWA MEM software that uses that does the alignment, but it actually is going to take a long time. It takes around, so for example, for this particular sample, for an exome sequencing sample, it will take close to a day, a little less than that maybe, but it's pretty long. It takes pretty long, right? So yeah, I mean, so for WGS data, which is like whole genome sequencing, the amount of time required will be much more. So it takes like 1.5 2 days to run a single pipeline or like a single alignment. But you can optimize this. That using that K value, you can change the parameters, tweak the parameters a little bit. Having a little bit less accuracy will get you the results in a much faster manner. Okay? So you can uh, uh, like consider the trade-off. Next, you get the SAM file. The SAM file I'll be able to show you. BAM is a binary file I won't be able to show. This is the SAM file. So this tech tells you this is the sequence. This is chromosome, which chromosome, and which is the lane number of that particular alignment. So it tells you where in the chromosome it has aligned to which lane number. So the UNs are your unknown alignments, which basically you do not know where it has aligned. But yeah, it'll it tells it these are your header files, header lines, and these are your basically alignment types. Right here, this is the alignment here given. So if you can see, this is the alignment, and these are your quality scores here, right here, from starting from F. These are your quality scores. So like this, as I showed in the last, sorry, this got stuck. As I showed you in the last PPT as well. So let's go back. Yeah. So yeah, it's exactly how it same looks like. So it, this header line is your basically telling you information about the alignment, position, sequence, and quality. And there will be other tags associated with this alignment, some more information associated with the alignment. So yeah, this is what it means. Oh, I think this just got stuck. I need to re-log in again. Just give me a minute. Sorry for the interruption, guys. I just tried. Yeah, I hope you can see this. Okay, so this was the SAM file that has been generated, which is after alignment with a particular reference file. Now, since you see this file size of the SAM, this is like 48 GB, close to 48 GB, right? But if you convert it to a binary format, the size significantly comes down. It comes down to almost 9 GB. So it's kind of like 1/5 of the SAM file. So that is the reason we converted it to a BAM to actually kind of compress the file and store it, because these files anyway, if you're dealing with hundreds of samples, you need to store hundreds of BAM files, and if you don't store BAM, if you try to store SAM, the space required will be 5 or 6 times more. So that's why we use the BAM file. We convert the SAM to the BAM. How do we do that? Is basically by using SAM tools. So this is the command that we use. So this is the SAM tools is the software. So it's also one of the most widely used softwares for uh, SAM BAM conversion and others, like other things it can do, FastQ to BAMgram, and can do mapping, it can do filtering of files and stuff. But SAM tools is one of the most widely used softwares in genomics across the world, right? Okay. So yeah, this is SAM tools. The command is view. uh, your This B is representing the output is BAM, and this S is representing that your input is SAM. Then you give the input SAM file, and you pipe it, and try convert it into a BAM file. So it's pretty simple. The SAMtools software is pretty fast. Does this conversion in less than an hour. It's like 30-40 minutes max. So this is actually pretty good. I have actually installed SAMtools through Conda, but you can use other ways to install it. You can use the binary and stuff you can do with that. You can also use Docker. So yeah. So so first you started with FastQC. We checked the we started with two FastQ files. We checked the quality. The quality was fine. We aligned the FASTQ files with the reference genome and converted and got the SAM file. Then we converted this to a BAM file, right? We are in at this stage. Now, after this, we'll do the variant calling, but the BAM file needs to be sorted for the variant caller to work properly. The variant caller will take a very, very long amount of time to call the variants, because it will sort it by its own, then it will try to call the variants. So it requires a sorted BAM file for its input. So we sort this particular BAM file. That too also we use SAM tools. So this is the command. So SAM tools sort is the command. So you do SAMtool sort, name of the file, BAM file, and you output it as a sorted BAM. And also, the variant calling softwares also expect there is an index to the particular BAM file. So SAMtools only indexes this particular BAM file. So the command is SAMtools index, and you give the BAM file. It will index the BAM file, and the file that will be created is a dot BAI file, right? So this is your sorted BAM file after SAMtools has worked on it, and this is your .bam.bai file, which is your index file, right? So you have sorted your BAM file. Now it's time for the variant calling. So for variant calling, we used deep variant in this case. So this is actually the one that I was telling about. So this is the GitHub page for this variant. It's actually developed by Google, so it's pretty good in terms of the computation stuff. And it can actually do a variant calling for a lot of stuff. It can do for whole genome, exome. It can do RNA sequencing case study. It can do RNA sequencing variant calling as well. It can do trio analysis. It can run on short read. It can run on long read. It can do a lot of stuff. So if you are interested in variant calling, this is a pretty solid thing to study, deep variant. So this is a kind of the state of the art currently. It uses some deep learning models to do stuff, but the idea is pretty much similar. It'll identify those particular regions of the genome where there are evidences of variant formation, and it will try to combine all this. But in this case, it does it through a yeah, what to say? It does this through the deep learning method, not an usual statistical method, right? So we use deep learning, we use deep variant to call the variants. So this is the command. So actually, deep variant installation of deep variant is a little bit of issue. So we installed it using Docker. So we have to run it using Docker as well. So the command is that when you run Docker, you need to specify your input directory and specify your output directory, then the command. So these are your input and output directories I have specified here. And this is the command, Google/deep variant. This is the command. And for model type, I have given as WES because mine is a exome sequencing data. So this is WES whole exome sequencing. Then I've put the reference, which is my reference FASTA file, GRCS38 reference FASTA file. My input file is a sorted BAM file, and it automatically takes the .bai index file with it. So I don't have to specify it again. And the output will be of a VSAF format, variant calling format, as I said. And I've specified the output file. And then it also requires an intermediate result directory where it kind of stores the intermediate result before finally giving you the output of the uh, most uh, confident variant calls, right? Let's look at the output, how it looks, right? So after you have run deep variant, it will give you a VCF file. So this is your VCF file. So I'll do a less because so yeah, as I was saying, in the first line, it'll tell you the VCF format. Then it'll give you all the information. What does pass mean? That means all the filters have been passed. What does ref call mean? It means that genotyping model thinks that this is a site for reference. What does low call mean? It means that confidence in this variant is being real is below the calling threshold. So these type of all the type of information that is provided inside the VCF file is explained in the header. So what it actually means, right? So you can read it in the other parameters as well. But the actual VCF file starts from here /chrome. So when you're trying to extract information from the VCF file, just remember it starts from this hash chrome. Before that, it's all header, right? So how it formats it. So this is the VCF file, the chromosome this is the position. We do not have IDs because I have not provided IDs for this, but we have reference, alternate. The quality you see, it's pretty low. It's zero point 0.1. It's not good at all. So it's given a ref call filter. So what was ref call? A ref call is that model thinks that this site is reference. So this is not an alternate allele, right? What it feels, right? So let's look at some pass variant. So this is a pass variant, right? Sorry, this is a pass variant. Chromosome 1, this is the position. The qual is pretty good. 30 is a decent qual. This is a pass variant, and along with that, it has given all this information. Now, what does GT mean? GT means genotype. GQ means genotype quality. DP means depth. AD means allele difference, and so on. Like, right, you can find all this information, all what each of it means. So, see, GT is genotype. GQ is conditional genotype quality. DP is read depth. Minimum DP is minimum depth observed within this block. AD is read depth for each allele, which is also known as allele depth. VAF is basically varial allele fractions, and every information that is given inside the VSAF, you will find that explained in the header. So what it means is basically, say for example, if you take this particular variant, so it is given in this format. So GT is the first one. So in the first delimiter, GT is like 1 1, which means your um, the genotype that has been called is a 1 1. It's a homozygous alternate genotype, meaning the variation is present in both the strands of the genome. GQ is 25, which is the genotype quality. Again, it's a Fred scaled quality. 25 is decent. It's a good one. What is the allele depth? What is the depth of the region? What is the allele depth? And rest of the stuff, everything is given like this. So let's take this example. So what do you think this means? Why is such a big sequence given here? What it means is basically this sequence has been converted to G. So it's a deletion. So this whole sequence was there, except from the G, rest of them has been deleted. So the alternate remain is only G. So this is a small deletion. This is less than 50 base pairs. So this is, you can consider this as a small deletion. It's not a structural variant, but yeah. So this is what it means. And this is a ref call variant. So this is not a pass variant like this, but yeah, this is how your genotype year will look like. basically your VCF file will look like after using some sort of VCF calling variant calling software. Now, the deep var or the deep variant software for variant calling calls for SNVs. We used Manta to call for SVs. Now, let's look at this. So in this case, I also installed Manta through Conda. You can I'll share the GitHub page if you want to take a look. So this software is developed by Illumina itself, so it works pretty well on Illumina paired in data. So you can take a look if you. Sorry. Yeah. Yeah. So here, so this is the Manta calling variant calling software. So this script comes with the software as well. So with the script, you have to give the run directory. If it is an exome or a WGS data, in my case, it's an exome. The reference file, this is the reference, and the BAM, the aligned BAM file, right? So this one you can see, and this is the log file that is getting created. So what in this step it does is it creates this particular Python file. So run workflow.py. In the next step, you need to run this run workflow.py to get the output. So let's look at how the output looks. So let's see. Uh, yeah, it's in this particular folder, I think. Yeah. So this, sorry, not this. This file is actually the file that gives you all the SVs. So this is also a VCF file, but the format is a little bit older compared to that. So yeah, it gives the reference file as well, which reference file that has been used, all this stuff. Now, if you see, the VCF file is very similar to the SNV VCF file that we saw, but there are slight caveats. So position and chromosome same as before, but in ID, it gives what it thinks the type of variation it is. It is a duplication, it thinks, and this is the reference given by G, and it's a duplication, so it doesn't give an alternate allele. So it gives like this tandem duplication. It's a pass variant. This is your end. So this was your start, 1485124, and this is your end. See how much bigger duplication this is. It is in chromosome 1. And this is the SV type, which is a duplication, and this tells you the SV length. What is the length of the SV? So this type of information is given in the SV VCF file. It also gives you the genotype if it is present in one strand or it is present in other two strands or not. So in this case, the genotype is a heterozygous genotype 01. FT is format is pass, it's good. GQ is a genotype quality, which is 98, it's pretty good again. This PR thing that I wanted to highlight here is the paired read. How many paired read are supporting this particular variation? How many split reads are supporting this particular variation? So this kind of information, what are the evidences for this particular variation to be called as confident? Those information is also given in the particular VCF files. So this is how the BCF files look like. It's a pretty big file like this. So in some cases, they'll give you the whole sequence. If it is small enough sequence to be fitted here, it'll give you the whole sequence in this case, deletion has happened. So this whole sequence has been deleted, right? And the genotype is 01, meaning it has been deleted from one strand. In the other strand, it is still there. So this is what the VCF files and the different variant calling files look like. And in the last step, after you've gotten, say, SNP VCF files, you've gotten SV VCF files. Now you want to see what these different types of files is actually what these different types of variants is actually meaning in the wider context. Now, so you use softwares like ANOVAR, as I was saying, ANOVAR, Intervar, and you try to annotate these files. So this, I think I should, once, give me a second, I'll download this file and show you. I think it'll open better in Excel. So this is also one Linux command that we use pretty regularly is basically scp. scp, and you give the place from where the file needs to be downloaded. This is the whole path of the file, and then you give the path where it should be downloaded in your own system. So in my case, downloads. It asks for the password, and then it downloads like this. So yeah, okay. So this after annotation, this is how the your data will look like. Just let me open this. Yeah. So this is how your data will look like. So it'll give you chromosome. Start end will be the same because these are SNVs. So this is one nucleotide change. What is your reference? What is your alternate? In which gene it is falling? Is it an exonic? Is it an intergenic? Or is it something else? So these are the categories that it gives. So if you filter it, so if it is downstream to a gene, if it is an exonic, it is if it is in its splicing regions, if it is in a non-coding RNA region, if it is in UTR3, 5, where the EA is falling, the variant is falling, and it also along with a lot of other scores. I'll not go into the details here. It gives you the clinical significance. So, for example, it tells you if the variant is pathogenic or not. So if you see, these are all the pathogenic variants that it has uncovered. It's not like very high. The total number of variants were like a million or so. Out of them, only 8 of them are pathogenic or likely to be pathogenic. And these are the genes that are associated with it. And these are the type of mutations that has happened. If it is a stop gain as in like prematurely, there has been a stop codon that has been inserted because of this particular mutation. Or non-synonymous. Basically, there has been a protein coding change inside the in the in your basically gene, but that is a non-synonymous variant. Basically, a particular type of protein, an acidic amino acid, has been replaced by a basic amino acid, stuff like that. And which genes are involved? So you can look into these particular variants, and you can check if these variants are associated with particular disease. If you find particular variants that are associated with particular disease in your data set, then you can have an idea if that person is at risk, or if that person is having the disease, or if that person is somehow prone to the disease or something like that. So this is how the annotation will look like. Same with SVs as well. That will also tell how many genes, because SVs are longer in size, right? If it is 1,000 or 2,000 base pair long SV, it will most probably cover more than one gene. So how many genes it is covering, what type of SV it is, and what is the clinical indication of that particular SV, what can actually happen with that SV in terms of the biology. Along with that, you get other stuff as well. Like as I was saying, you get this OMIM are reference numbers. So OMIM is a disease database. So these are your different disease IDs that are associated with these particular variants that you can go and cross check and see if these are actually uh, like associated with like rare diseases or what type of diseases they're associated with. And these at the end are telling you if the particular variations are heterozygous or homozygous. So if it is heterozygous, it's most probably it could be de novo as well. It might have been not been passed on from the previous generation. It might have been happening in your lifetime as well, or it has been generated in your genome, but it has not been passed off on from your parents. So that is what a de novo mutation is. It's a new mutation like that. So these kind of information you can get. So yeah, that's all for for my side. Now let me go through the questions. Okay. Okay. Sorry, I can't share the data set because this is a patient data set and like I'm not supposed to share it. But what I can tell you is basically where you can get FASTQ files. So you go to 1 kgp, you go to just type it like this 1 kgp FASTQ files. You can go to this link And just a second. So there are these databases, which is one is SRA. You can try this, European Nucleotide Archive, and another is you can find it here as well. Just wait a second. Yeah, in this data portal, I'll give you this link. So in this one, they have 1,000 genomes. Like it's basically what it means, 1,000 individual data you can have. So they have all given this information. What is the sex? What population is the data from? So you can go in any one of these, and you'll find the FASTQ file here. Read1.fastq. This is the read2.fastq. There are multiple fastq files in these for like more than 3,000 individuals. So if you want to practice this alignment or different types of these pipelines on fastq files, you can download it from 1 kgp. And I'm extremely sorry I won't be able to give this particular fastq file because this is a patient file, so I'm not supposed to give out anyway. Right. And yeah, PPT, I'll record this lecture and upload it so you can check it there. Yeah. Docker. Okay, Docker. Uh, how should I explain? So, Docker is kind of, you can imagine, if you are familiar with uh, so if you're familiar with things like Conda, do you know what Conda is? It's basically, it creates an environment with a particular softwares and the particular tools required for a particular analysis or a particular program to run. Similar is a concept with Docker. You can just download the Docker image. It comes in the form of an image. It's not an image. It's not a JPG file. It's a Docker image. So once you get the Docker image, if you install the Docker and run the Docker image, it will create a virtual environment inside which all the available tools for that particular software will be available. You don't need to worry about anything. You just give the input, you give the output, and whatever commands that there is. But rest of the stuff, all the dependencies, all the different types of, say, for example, you require Python 3 and your system has Python 3.8. So it will create a virtual environment where there is Python 3 and your software can run like that. So yeah, so Docker is something I also am not very fluent in terms of like understanding what Docker is. It's like it's a new thing for me as well. But generally, you might want to know that this is just a virtual environment that is created inside your machine, which can provide you for all the dependencies and the tools required for a particular software to run. So yeah, that's all. Yeah, do you have any more questions? Okay, I hope you're asking about splicing mutation. I'm not sure what you have written there, but I think it's splicing what you mean. So splicing is basically the mechanism by which DNA, from DNA comes RNA, then protein, right? So in eukaryotic cells, from when the RNA polymerase get transcribes the mRNA from the DNA, it is in a pre-mRNA form. It's in a nascent form. That mRNA needs to be processed to get the ultimate mature mRNA. So that process of converting a particular stretch of an RNA, removing the certain parts of the RNA, which are the introns, and joining the rest of the parts of the RNA, which are exons, that process is known as splicing. Now, this splicing is very specific in nature. In specific areas of the mRNA, the splicing will occur. Now, if in that region of the mRNA or the genome, basically where the splicing mechanism will come and sit and cut the mRNA, if in that region, say for example, there is a mutation, so what will happen is the machinery, which is the spliceosome, it won't be able to come and sit on the mRNA and cut the mRNA. So that will lead to defective splicing, which will not form a complete or a good protein downstream. So splicing certifications, I just got mixed up in this. So splicing mechanisms get affected if there are mutations in the area where the splicing mechanism will act on. So those are mostly splice mutations. Right? Certification, from what I know, is yeah, you are supposed to get. IEES will contact you. So I have forwarded all the. There were 234 people registered for this particular course. I have forwarded all the information to the IEES admin. They have created a profile for me where I can where I can kind of upload these lectures as well there. So I think they'll contact you and like they'll send out mails and stuff like that. Yeah. So 234 people who have registered, I have forwarded their emails. Yeah. Okay. Any more questions? No, these two were only there. So like 2 hours, 2 hours, 4 hours. That was the schedule. Actually, I have not like kind of. If you want to really cover a course on NGS analysis, including RNA sequencing analysis and everything, it takes around 3 months on a minimum. So uh, it's I have not covered many topics, obviously, but I just wanted to give you an understanding about how these things work. That was the primary objective for this course. Yeah. Yeah, you can perform variant calling on your laptop depending on how what the specifications are, and depending on how big your files are. So if you are, say, for example, are taking microbial genome data, so it's going to be much smaller, right? So it other type of uh, yeah, like species data where the genome size is pretty small. So in those cases, it's possible to run on your laptops. But since we deal with human data here, so it's not possible to run human sample pipelines on normal laptops. Okay, then we are a lot above time. It's 7:35 now. I think I'll close the session here. Thank you all for joining. It's been a pleasure on my side to teach this. I love teaching this part for 2 days. Very, uh, it was a very enriching experience for me as well. Yeah. Thank you everyone who joined, and I'll leave my email ID here if anyone in the future wants to get in touch. Do email me, and yeah, thank you all for joining. Thank you.